Scientists are aware that prolonged space travel can affect astronauts' physiology and cause health issues. Microgravity can cause muscle atrophy, bone density loss, and vision issues. In addition to causing the brain and cerebrospinal fluid to transfer, it may be associated with brain damage. 5 astronauts had elevated levels of proteins in their blood, which is typically observed in patients with head injuries and neurodegenerative diseases. Welcome back to my channel. Prolonged space travel may lead to brain damage and cellular decay. Over the past several years, scientists have published research suggesting that prolonged space travel alters the minds of humans. These studies were initiated because astronauts returning from lengthy missions experienced vision problems and swollen optic nerves. Now, scientists are pondering whether these protracted space journeys cause brain damage. In a new study involving 5 male cosmonauts, researchers analyzed blood levels of various proteins that are frequently observed in patients with head trauma or brain disease. They discovered that, on average, the cosmonauts' protein levels were higher 3 weeks after the mission than they were before. There is a need for additional research to determine whether or not these changes are clinically significant. However, the ongoing research exemplifies the types of tests that need to be conducted to better comprehend the impacts of the brain's changes during long-duration spaceflight. Humans have visited the International Space Station for more than 2 decades, which orbits Earth more than 300 miles above the planet. Typical trips last approximately 6 months. Since the space station is in free fall around the Earth, its inhabitants experience near weightlessness. This microgravity is believed to be the primary cause of a variety of changes that the human body undergoes during space travel, including the loss of muscle and bone density. In the past few years, brain imaging has disclosed a decrease in the volume of gray matter, which contains neuron cell bodies, and an increase in the volume of cerebrospinal fluid. The research team that uses imaging to study the effect of spaceflight on the brain has published research indicating that in some people who have had long missions on the International Space Station, the brain has shifted higher in the head towards the top of the skull, and cerebrospinal fluid occupies a larger volume below and in the center of the brain. To search for signs of brain damage, scientists measured the levels of 5 distinct proteins in the blood of 5 male cosmonauts before and after approximately 6-month space station missions. These proteins are biomarkers that can reveal the condition of the brain without opening the skull. The cerebrospinal fluid in the brain and spinal cord is the best fluid for researching biomarkers, but obtaining it requires an invasive spinal tap. Blood is the second best biological fluid for testing the health of the brain, and its collection is significantly less difficult. The biomarkers identified in this study can be used to infer the extent of neurodegenerative or traumatic damage. For instance, the neurofilament light chain is a structural protein that maintains the axons of neurons, which transmit signals to other neurons. In a healthy individual, there should not be a significant amount of these structural proteins in the blood. However, if a person has a neurodegenerative disorder, the proteins can leave the neurons and enter the circulation. 20 days prior to launch, the researchers measured 5 proteins and calculated the average level of each protein among the 5 cosmonauts for each protein. This was then compared to the average level 1 day, 1 week, and 20 to 25 days after the cosmonauts returned to Earth. 2 of the proteins had elevated concentrations both 1 day and 7 days after the missions. These levels decreased over the next 2 weeks, but remained above the cosmonauts' pre-mission baseline. A third protein was not substantially elevated in the first week after returning, and had returned to baseline levels after 3 weeks, showing that it was not indicative of possible brain damage. For the last 2 proteins, scientists typically examine their respective ratios. After the return of the cosmonauts, the ratio decreased, a phenomenon sometimes observed in patients with neurodegenerative diseases. It was remarkable that some levels remained elevated for the entire 3 weeks, and the results confirmed brain damage as a result of prolonged exposure to microgravity. Clearly, This is a pilot study, but the data quality and analytics are so solid that I have no doubts about the overall impact. The severity of these effects remains undetermined. Initial data suggest that there is some form of brain injury, but more research is required to determine the effect on astronauts' health. This is not conclusive evidence that those who travel to space will develop Alzheimer's disease. Future studies measuring the biomarkers for longer than 3 weeks after astronauts return, as well as while they are on the space station, would help determine if the elevated levels are due to time spent in microgravity, as opposed to the change in gravity upon return or the intense force experienced during landing. Understanding the cause of the elevated biomarkers would allow space agencies to determine the most effective countermeasures. Since imaging studies have shown that brain changes do not occur during shorter trips, it is most probable that the elevated levels begin while astronauts are still in space. If biomarkers are only elevated for a few weeks, this may indicate a significant reparative process. And other studies have demonstrated that brain alterations and cognitive effects persist for at least a few months after spending months in space, suggesting that elevated biomarkers may persist for longer than a couple of weeks. Another study has shown that an enzyme in the bacterium E. coli made more errors copying synthetic DNA when exposed to zero gravity than the same enzyme did in normal gravity. The possibility that some enzymes function differently in space than on Earth suggests that enzymes involved in maintaining the integrity of our DNA, such as polymerases and others, may be affected by spaceflight. This finding necessitates additional research into other enzymes that are involved in essential life-sustaining pathways. Scientists are aware that prolonged space travel can affect astronauts' physiology and cause health issues. Microgravity can cause muscle atrophy, bone density loss, and vision issues. In addition to causing the brain and cerebrospinal fluid to transfer, it may be associated with brain damage. It is still unclear the effect microgravity may have on DNA. DNA polymerases, a class of enzymes that copy DNA and restore damage, are the subject of research. Polymerases read the arrangement of the 4 types of nucleotide bases on the original DNA strand, and position the corresponding base on the corresponding section of the new strand. However, these polymerases are imperfect, so some have affixed proofreading enzymes that double-check their work and identify errors. Researchers wanted to determine if polymerases might produce more or fewer errors in microgravity, which could have implications for how radiation damage in space is repaired. Some microgravity experiments are conducted in space at locations such as the International Space Station. It was logistically simpler to conduct his experiment closer to Earth, aboard a specially modified aircraft, called the Vomit Comet, that obtains zero gravity through a series of rapid ascents and descents. Astronauts began training on an aircraft that simulate weightlessness by performing roller coaster-like maneuvers in the air since 1957. The simulation causes nausea in some passengers, hence the appellation Vomit Comet. In 1973, NASA assumed responsibility for the Air Force program that had preceded the space agency's formation. Since 2008, Zero G Corp has been responsible for the training. Although the program was designed for astronaut training, it is not exclusive to prospective astronauts. Scientists, engineers, students, and celebrities have all experienced weightlessness on the Vomit Comet. Its contents experience a fleeting 20-second period of weightlessness as the plane flies 10,000 feet above the Earth's surface and reaches the peak of its parabolic flight path. Science team on board the plane made the experiment. When the plane attained zero gravity, a mixture containing synthetic single-stranded DNA was injected with E. coli polymerase. As the aircraft departed from zero gravity, they injected a second mixture to cease the reaction. Back on Earth, the data indicated that the polymerase with the proofreader affixed made marginally more mistakes on the Vomit Comet than in normal gravity, although this effect was not always statistically significant. Some polymerases lack proofreading enzymes, indicating that the proofreader was able to remedy some of the additional errors made by the copying component when it was not attached. The research has no direct bearing on astronaut health during extended spaceflight. Since the experiment employs a polymerase found in microbes, and the DNA was exposed to zero gravity for only 20 seconds, the findings are extremely different from what astronauts actually experience. The E. coli polymerase utilized in the experiment is a member of a family of polymerases that accounts for a small portion of the 14 known DNA polymerases found in humans. If certain polymerases are affected by microgravity, this may have implications for astronauts' telomeres, which are DNA sequences at the extremities of chromosomes that are believed to be associated with human aging. A type of polymerase known as telomerase duplicates telomeres. They investigated how spaceflight affects telomere length, and discovered that there is some support for the notion that microgravity could affect telomerase activity. Astronauts are also exposed to radiation from the Sun and distant galaxies while in space. Radiation sort of behaves like a bowling ball, whereas DNA represents bowling pins. These radiation particles move so quickly that they can separate DNA or induce mutations through a variety of mechanisms. This DNA damage may increase astronauts' susceptibility to cancer and degenerative diseases. Going into space, aside being a dangerous endeavor for the risks involved just attempting to go there, a successful space mission came with its own strings attached. Brain damage and cellular decay may be on the plate, further studies will confirm that. The space agencies already are trying to mitigate the risks associated with exposure to microgravity and space radiation. Thank you for watching.